everyone. Welcome to the Psychonomic Society's One World Cognitive Psychology Seminar Series. I'm really excited today. We have Dr. Penny Lewis from Cardiff University presenting. Dr. Lewis is an expert on sleep, memory, and mental health. Her research team examines how the sleeping brain processes information and how we can use sleep to improve memory, enhance creativity, and improve mental well-being. So I'm really excited to learn from her today. As a reminder, you can ask questions using the Q&A feature, and at the end of the talk, we'll have some time for moderated Q&A. But with that, I'll let Dr. Lewis take it away.
Thank you so much, Lisa. It's a real pleasure to be here, and I'm really grateful for the invitation. I'm just going to share my slides and make them as big as I can for you there. Hopefully you can see them clearly now. So yeah, it's a pleasure to be here, and I'm going to talk to you today about sleep engineering and how we can use it to enhance health and cognition. And I always like to start my talks by acknowledging the people who actually did the work. So here you see PhD students and postdocs in my lab who did this work, and I'll introduce them each as we come to their part as we go through. But let me start by introducing sleep. So this is something that we're all intimate with, and it's probably near and dear to all of our hearts, but you may not have all thought about the fact that as human beings, you actually spend roughly a third of your life asleep. So it varies, of course, between people, but roughly 8 hours a day. And that's more time than you spend doing any other single activity. So just that should suggest to you that sleep is important for something. Then if we look across the animal kingdom, we find every single kind of animal that's been examined exhibits something that we would consider a form of sleep. And many of them have actual specific adaptations to allow them to sleep. So one of my favorites is dolphins. Of course, they're mammals, and that means they have to breathe, even though they live in the water. So how do they handle that when they sleep? Well, they've evolved split brain sleep. So they sleep with just one hemisphere at a time. And the hemisphere that's awake keeps them swimming, and they can swim in big spirals and come up for a breath and swim down again. So that's a clear adaptation specifically to allow them to sleep. And then my absolute favorite is actually animals that hibernate. So I put a bat on here. So everyone will be aware that, of course, hibernation is a way of saving energy when there's not much food around. And one of the things that happens in hibernation is that the core body temperature really cools down. Well, it turns out that if your body temperature cools down too much, then your brain can't obtain sleep-like activity. So hibernating animals actually periodically warm up. So they're investing energy just in order to sleep. So it's another really cool adaptation that allows these animals to get the sleep that they clearly need for something. So what happens when we sleep? Well, sleep is all about the brain. These are a couple of my undergrads who were having lots of fun and measuring sleep with electrodes, and this is how we tend to do it in the lab. We put little electrodes all over the scalp and on the eyes and on the muscles under the chin as well. And what we find with this is that as we fall asleep, we go through what we call stage 1 sleep. This is a pretty brief phase of sleep, a transition phase. And basically what happens here is that the oscillation frequency of your EEG just slows down a little bit. So it looks a bit slower, quite similar to wake, but just a bit slower. This is, EEG, if you're not hearing or experiencing any specific stimuli or moving or doing anything particular, this is just kind of resting EEG, and then it slows down a little bit. And then if you move from stage 1 into stage 2 sleep, this is actually the dominant stage of sleep that it dominates most of the night. And it's a little bit deeper than stage 1. It's no longer a transition. The basic oscillation frequency slows down again a little bit, and you start having these bursts of high frequency activity called sleep spindles. And these occur over localized areas of the cortex, so they don't occur everywhere at once. And I'll point to it again. I think I've put a red circle around it later because I want to really emphasize these sleep spindles to you because they're quite important. There's something we're going to come back to. But if you stay asleep through stage 2 sleep, then you'll move into an even deeper stage of non-REM sleep that we call slow wave sleep. And here the EEG does something really fantastic. It starts to exhibit these huge slow oscillations that you can see here. And by the way, the spindles occur in this stage as well. They kind of ride on the top of the slow oscillations, but you can't see them because of the scale, because they're so tiny in amplitude compared to the huge amplitude of these slow oscillations. This is caused by mass neural synchrony. So millions of neurons, mainly in the cortex, all firing at the same time, causing a peak in electrical activity, and then pausing, causing a trough, and then firing again. And the frequency is a bit slower than once per second, so about 0.8 hertz. And I mean, I think this mass neural synchrony is really interesting because it's so different than what you see in any other state of consciousness, except some kinds of deep anesthesia. You'll see this. And then if you stay asleep, you'll move into the sleep stage that's maybe the rock star of sleep that everyone's heard of, rapid eye movement sleep, or REM. And as sleep scientists, we actually call this paradoxical sleep. And it's paradoxical because it actually looks very similar to wake. So all of that neural synchrony is lost. Now all your different areas of your cortex are kind of doing their own thing. So when we add it up and look at it on one electrode, it just looks kind of noisy, like stage one or like wake. But you're still deeply, deeply asleep in REM. So that's why it's paradoxical. And of course, you have rapid eye movements under closed lids, and that's how it gets its name. So those are the stages of sleep. And if we just, and I just wanted to emphasize the spindles again. And if we just look across a night, this is a cartoon of a hypnogram. And a hypnogram is the way that we sleep scientists tend to plot our data across a night to show what stages people were in. And what we tend to put REM up here by wake, and then you've got stage one, stage two, and slow wave sleep is kind of broad in this. And what you can see here is two things. Firstly, people tend to cycle through these stages, all of these stages. So stage one, stage two, slow wave, a little bit of REM. And then again, stage one, stage two, slow wave, a little bit of REM. And that cycle repeats again and again and again across the night, about 4 to 5 times across the night. There's a huge amount of variability in this. And what you also can probably see here is that the early part of the night is dominated by longer epochs of slow wave sleep and very little, if any, REM in each cycle, whereas the late part of the night is dominated by longer epochs of REM in each cycle and very little, if any, slow wave. And so we can kind of almost think of this as a kind of a physiological drive to get slow wave sleep first, like a hunger. You need to get slow wave sleep first. And then REM is kind of the dessert that comes later, but you still have to keep this cycle pattern. The brain goes through the stages in the correct order. Okay, so that's the structure of a night of sleep. And hopefully, you know, I've convinced you that sleep is important because we've got these adaptations and that it's something, the brain, it's something about the brain. Sleep is all about the brain. The brain doesn't just switch off when we sleep. Instead, it's going through this very intricate architecture of quite different stages in a quite precise order. So sleep is important for the brain. Okay, so what is it actually doing for the brain? I'm not going to be able to fully answer why we sleep in this talk, but I'm going to touch on 2 different kinds of functions and what sleep engineering can do for those kinds of functions. And so the first thing that I want to touch on is sleep for restoration and repair. Now, here's a nice, healthy, plump brain, and we'd all like our brains to look like this. I'd love it if my brain still looked like this. And here's a cartoon of nice, healthy, slow oscillations, which we'd all love to get every night. But actually, we don't. So we know that many times our sleep isn't very good and it can break down and fragment. And one of the cases when this is the most obvious is just aging. So as we age, our sleep gradually changes and breaks down. And one of the ways in which it changes is that we gradually lose those slow oscillations. So that mass neural synchrony goes away. And we don't know why, but one possibility is that the brain gradually becomes less able to exhibit that synchrony, that it's actually quite a tough task to have that degree of synchrony. And so as we have gradual atrophy, and cell death and all the other things that happen as we age, it becomes harder and harder. So in most women most men by the age of about 60, those slow oscillations are completely gone. And in women, it's about 10 years later. So here's a picture of a brain that has suffered from atrophy. And here's the data that links the loss in slow oscillations to the atrophy. So why does the atrophy matter, and why do the slow oscillations and the fact that we lose them matter? Well, it's been shown that the extent to which those slow oscillations have been lost across age can predict cortical atrophy in some regions, particularly some of these frontal regions that are shown here, and you can see in the bar graph. So these kind of data, along with other data on the aging process, convinced myself and many other sleep researchers around the world to try and think about ways that we might be able to boost those slow oscillations and potentially try and slow down some aspects of cognitive aging, including atrophy. So that's the goal. So I'm going to tell you now about the first kind of sleep engineering that we're going to touch on in the talk. So this is called closed loop auditory stimulation, and it's a technique that was developed by Hongi Ngo, who was then in Tübingen in Germany. And basically what happens in this technique is very simple. He simply hits the brain with a sound. So these yellow arrows are meant to represent a sound, and the sound is just a patch. It's 50 milliseconds of pink noise, so it sounds like that's what the sound is roughly. And so it's closed loop because the way you do this is you monitor the EEG online as someone's sleeping, and then when they enter slow wave sleep, you predict when the oscillation will be coming up towards a peak, and then you try and play a sound right as it's coming up towards a peak. And what that does is it deepens the next trough and heightens the next peak and the next trough and the next peak. And if you do this again and again through slow wave sleep, you can increase the slow wave power for that night. You can sometimes extend the amount of time spent in slow wave sleep, and you can also boost some of the benefits that happen across that night of sleep as well, in particular memory consolidation. We'll come back to that later on. So here's some of the data that showed this. Here, basically, this is data from healthy young people. And on this dashed line here, this is where the sound was applied. This is an average across all of the slow oscillations for these people. I believe there were 11 people in this study. For all of the slow oscillations in their night that were stimulated for all of the people are averaged here. And you see the green is the stimulated average, and the black is the sham. So in the black, it's an oscillation that could have been stimulated, but it wasn't. So that's the control. And so basically, hopefully what you can see here is this really nice effect of the stimulation where it's really boosting these oscillations. Okay, so this was in Hongi Ngo's original paper, but then a student of mine, Jules Schneider, shown here, got interested in this question of could we use this stimulation in older people in order to try and boost their slow oscillations. So if we could, of course, this would be great because it would be a completely non-invasive, non-pharmacological way of doing this that can be delivered in the home. And so what she did is she actually went over to Germany and worked with Hongi and his lab, and they used exactly the same protocol on a group of people with mean age 56 years. And what did she find? So here's their data. And you see the stimulation group average is in red, and the sham is in black again. And the significance is shown with this black bar at the top. And what you can see here is there is an effect. I mean, there is a benefit of this, but it's really kind of disappointingly small. I mean, it's really tiny compared to what we saw in the younger group. And so this result, and I can tell you that in this older group, there was no benefit to memory consolidation either, whereas in the younger group, there was a benefit. They consolidated memories better if they had this stimulation. So, excuse me. Okay, so this was kind of disappointing. So it led us to think, all right, if this is going to work in an older group of people, if this form of sleep engineering is going to be useful in this older group, we're going to have to improve it a lot. So what we did is go back to the same data and do a lot of analysis to try and work out how we could optimize the stimulation. And the first thing that we did was simply look at when is the window on the oscillation if we play a sound when we're going to get an actual benefit on the next oscillation. Is there a window of opportunity? And what you can see is that indeed there is. So there's a nice window here in the healthy young people. And there's also a window of opportunity in the middle-aged group, but it's just much narrower. So this suggests that if we want this to work in even late middle-aged group, not even old, then we're going to have to target it much better. So we're going to have to be much more precise about when we deliver the sounds. So that's useful. And then the other thing that we did here, and I don't think, I didn't put in a slide for it, but the other thing that we did here is we looked at different kinds of slow oscillations. So slow oscillations are not equal. They're generated by different parts of the brain, and they can travel across the brain in different trajectories. They can have different slopes. They can have different kind of amplitudes, all kinds of differences. And so what we did is get all of the features that we could for the slow oscillations and use those to see if we could predict which ones would respond well to stimulation in terms of whether the next oscillation exhibited advantage. And we were able to predict that very well. So with about 80% accuracy, we could predict which oscillations we should be stimulating. So with that information, we're now ready to go back into the middle-aged group again. We haven't done it yet, but just try and do this in a more targeted way and see whether we can get more benefit. And what I can tell you is that in the meantime, other labs around the world have also been working on this. And I know of 2 papers that have come out in mild cognitive impairment patients showing that this kind of closed loop auditory stimulation is beneficial for their memories, essentially showing correlations between the extent to which the slow wave power is boosted and the extent of their memory consolidation across that night of sleep. So even in a patient group like that, it can be beneficial. So I think the message on this first kind of sleep engineering is it's promising. It needs work, and it's something that quite a few research groups around the world are working on at the moment. Okay, so I'm going to shift gears now to talk about another function of sleep, which is memory. And we've already kind of alluded to this a couple of times, but now I'm going to kind of dig down. And I want to start by trying to convince you that sleep is incredibly important for memory consolidation. And I always like to do this by getting people to actually do one of the tasks that was used in a really seminal study of this. So if you can all hold up your non-dominant hand, of course, I can't see you doing this in this forum, but I'm just going to trust that you're all doing this in front of your computers. And you can see that I've numbered your fingers 1, 2, 3, 4. And what I want you to do is press your thumb against your fingers in the order 4, 1, 3, 2, 4. 4, 1, 3, 2, 4. 4, 1, 3, 2, 4. 4, 1, 3, 2, 4. Okay, we can stop. So I think you get the idea. This is an actual task that was used in one of Matt Walker's studies back in around about 2002. And what they did is they trained a group of people on this task, and they basically had to practice this sequence 4, 1, 3, 2, 4 until they weren't getting faster anymore, until they plateaued. So people would get faster and faster, and they're going to get a bit fatigued, and at some point they won't get faster anymore, and that's when they stopped. And then they were given a test, and the test was simply 30 seconds. How many 4, 1, 3, 2, 4s can you produce? And you see here a cartoon of their data for simplicity. This was done around 10 a.m. And people produced roughly 21 4, 1, 3, 2, 4s in 30 seconds. Then they were sent home, told not to practice, come back 10 p.m., another 30-second test, and they produced roughly the same. No significant difference there. Then they were sent home again, and guess what? It was nighttime, so they slept overnight at home. And they came back the next morning, 10 a.m. They were given another task, 30 seconds. And what do you think happened? I'm not going to wait for people to respond because I don't know if you can respond on here, but I wouldn't be telling you about this if they didn't see an improvement, right? So there was a dramatic improvement in how fast people could do this 4-1-3-2-4 sequence across a night of sleep. And the extent to which that improvement occurred was actually predicted by the sleep spindles that occurred over the cortex controlling that non-dominant hand. So if it was the left hand, the right motor cortex, the spindles that occurred there. And this is part of the reason that I wanted to emphasize the spindles. There's bursts of high frequency activity that occur in stage 2 and slow wave sleep seem to somehow be important for this effect. Now, of course, because it was an academic paper, they were criticized and people said, oh, you know, it was just because that was the 3rd session and that's why they got faster. It was nothing to do with the night of sleep. They had a control group. The control group started 10 p.m., had the night of sleep, showed the improvement between the 1st and 2nd session, and then didn't improve across the day. So it really is about the sleep and the spindles. Seemingly something is going on here. So this improvement was up to 20%, and it suggests that at least some kinds of memory can really benefit from a night of sleep, and something's going on with those sleep spindles. Okay, so what is that? Well, fast forward 8 years, fast forward more than, 23 years till now. And I think we can finally answer that. I think we can say with confidence that the benefit that was conferred by the sleep is actually due to a phenomenon called replay or reactivation that happens during wake as well, but it also happens during sleep. And essentially what's happening is the brain is practicing. So the brain practices things that were recently learned or that are particularly salient or that were rehearsed recently while you're asleep. And some of the earliest data for this, or the strongest initial data, came from rodents. This paper was 2010, but people were actually working on this in the 90s. So the idea of replay has been around for quite a long time. And the way that it was measured back then, mainly in rats, was by looking at their place cells. And you will know that place cells are cells that fire in a particular location. So the cells in the hippocampus area, CA1, that fire in a particular location. So if you have a rat running down a track, then as it moves through space, there will be different place cells that fire in different locations on this track. So here you have cell A, cell B, cell C, cell D, like that, etc. And if you measure, if you monitor the firing rates of these cells in sleep before the rat ever experienced this track, and then again in sleep after, what you find is that there's a significant increase in the probability that those cells will fire in the correct order, as if they were running down the track after the rodent has trained on that track. And the reason that place cells were used for this is because it's a really nice, clear measure of practicing, replaying something that happened to that animal during wake. So that you see this more often after they've experienced this than if they've never experienced it. Okay, so that's where the idea of replay came from. But since then, there's been a lot of work on replay in humans. And I'm going to tell you about one of my favorite studies of this. And this is, again, quite a seminal one from Philip Pignot's lab. And what they did here is they got people to navigate. So they're playing a game, a video game. So they're navigating around a virtual environment. And this picture is actually the aerial view of the virtual environment. And they're doing this in the MRI scanner. And so they had to do this, for a while to get used to the environment. And they were given tasks like get from point A to point B in the environment as fast as possible. And obviously, the more they learned their way around, the faster they were able to do that. And their brain activity, their fMRI measured brain activity during the task was what you would expect. So, some areas of parietal cortex, some other areas, and critically, the hippocampus were active during this task, some visual areas as well. Now, that's not particularly exciting, but what is exciting is that they then got these people to go back in the scanner and sleep. and they collected brain activity, fMRI measures of their brain activity while they were in slow wave sleep. And they also had a control group, a different group of people who did everything the same, except that their task didn't involve any spatial learning. So they had a video game task with no spatial learning in it. And those people also slept in the scanner, and the data was acquired in slow wave sleep. And then they compared them. And what they found is that for the people who did this navigation task during slow wave sleep, the hippocampus was significantly more active than for the people who did the non-spatial learning task. And furthermore, they woke those people up then, the next morning. They got them to do the task of getting from A to B as quickly as possible. And they found a correlation between the overnight improvement in how quickly they got from A to B and the extent of activation during slow wave sleep in the hippocampus. So it's true that it's only 6 people, and these days you might have trouble publishing that, but I think this is a really beautiful early illustration of memory replay during slow wave sleep in humans, and also how it actually predicts behavioral improvement. And that's what we're talking about. We're talking about how the brain practicing during sleep could be the thing that facilitates this kind of overnight consolidation, memory improvement. Okay. But the plot thickens. I haven't mentioned sleep engineering yet with respect to memory. And so now I'm going to. So it turns out that we don't have to just sit around and wait for the brain to practice the things that we're interested in, the memories that we want during sleep. Instead, we can actually control which memories get practiced during what stages of sleep. And we do this using a technique called targeted memory reactivation. And it's very simple. The way it works is we pair sounds or smells or even tactile stimuli, but most commonly sounds, with some learned information during wake. And then we play those sounds back during sleep very softly, hoping not to disturb the participant, and it triggers reactivation. So if I show you this picture of a cat in this screen location, and I pair it with a meow, and I show you a dog in this screen location, I pair it with a woof, and then tonight you're sleeping in the lab and I'm there monitoring your EEG in the next room. When you go into slow wave sleep, if I play meow, your brain does cat. That's the idea. So we're triggering the brain to reactivate memories during sleep. And the reason I use the cat dog screen location example is because that's the task that was initially used to show this with sounds. And since then, the result that if we then check how well you remember those screen locations the next day, we find that you'll remember the cat better than the dog that wasn't reactivated in sleep. That's been replicated in many labs around the world now and in many different patient groups and different ways. So we absolutely know that behaviorally, there is a benefit of triggering these things in sleep. But what we didn't really have strong evidence for a while was the fact that this was actually triggering reactivation. So we kind of, as a community, we believed that people were better because it was triggering reactivation. We knew reactivation was a thing. But in my lab, we got very interested in this question of, can we actually show that this is triggering genuine reactivation? So Mo Abdullahi, who was then a PhD student in the lab, asked, can we detect memory reactivation after the targeted, TMR is targeted memory reactivation, so those sounds, after those cues, and can we do this in non-REM and REM sleep, both of those stages of sleep, using EEG classifiers, because we typically have EEG data when people are asleep. And the way he looked at this was using a task called the serial reaction time task, SRTT, which is quite simple. Basically, participants have to implicitly learn a 12-item sequence. So we're cueing them to produce this sequence using 4 fingers. So there are 4 fingers, but it's 12 items. Each finger is associated with a sound and a picture and a screen location. And so when they see the picture pop up in that screen location, they hear that sound, they know they need to press that button with that finger. And they're getting cued to produce a sequence that repeats again and again, but we never tell them there's a sequence. And amazingly, most people don't realize, certainly not during training, that they're doing the same sequence again. But they do get faster and faster at the sequence. And this is a task that we know is sensitive to sleep. If you don't mess with the sleep, people get faster, and it's predicted by the amount of slow wave sleep. We know that if we cue that task in slow wave sleep by playing the sounds back, that gives a benefit. And so Mo decided to look at whether we could actually detect the reactivation. And so what he's trying to do is detect which hand is being cued off by each cue. So how did he do this? He trained people on that serial reaction time task. Actually, let me back up. He didn't train them straight away. First, he got them to come into the lab for an adaptation night. So they've never done the task. And during that night, he played the sounds to them in the correct order for the sequence, exactly as if he was applying targeted memory reactivation. But at this point, the sounds are meaningless. They don't mean anything to the participants. Then the next day, or in the next couple of days, they came in and they trained intensively on the task with the sounds and the pictures and the button presses and everything. And then they stay another night in the lab. And this time, when the sounds are played during slow wave sleep, the sounds are meaningful because they're paired with the task. And so they should trigger reactivation. And they should trigger reactivation of a particular finger, but it's easier for us to check for reactivation of which hand, because we've got 2 fingers on each hand that we used in the task. So then what he did is take the data, the wake data, to train a classifier. So that's essentially a fingerprint of what the EEG data should look like when they are actually doing the task with each hand, and then apply that classifier to test for each trial after each sound that was played in sleep. Could we classify? Can we tell which hand is being cued? And it did take him about 2 years to get this to work. But what I can tell you is that, yeah, it did work. So we were able to classify above chance which hand is being cued when this is done during slow wave sleep. And here's what the data look like. So the adaptation night is shown in red here, and the experimental night is shown in blue. And what you can see is that the sounds are, this is where a sound is being played. And so if you had a reactivation, it should be happening right in here. And what you can see is that there are 2 places where the experimental night classification, the y-axis is the AUC, the classification accuracy, is significantly different for the red and the blue. There's an early one and a late one. And so this is really nice because it not only shows that we can detect this, but actually there could be 2 different kinds of reactivation, kind of early and late, and that's been substantiated by later studies as well. Okay, so we were super excited about this when we did it, but we also still had the question of whether we could detect this reactivation in REM sleep. And the reason we were worried about that is because 90% of the literature on targeted memory reactivation is in non-REM sleep. So people don't tend to stimulate REM sleep, but we were really interested in stimulating REM sleep for reasons that will become apparent later on. And so we needed to know whether our stimulation was actually causing reactivation. So we tried doing exactly the same thing. We had exactly the same task, the same protocol. And what I'll tell you is that initially we couldn't do it. We were super frustrated. So when we trained the classifier on wake and tried to apply it on the REM data, we couldn't classify. And at some point we shelved the whole project for a couple of years because we didn't know what was going on. But then we looked back into the rodent literature, and there was actually only 1 paper in the rodent literature where they had identified place cell reactivation in REM sleep. And this is this Louie and Wilson. And when we looked at it, and we talked to colleagues who work on rodents, we realized that they were doing this the other way. They were training the classifier in sleep and then applying it back onto the wake data. And by doing that, they were focusing on the features that actually were there and identifiable in REM sleep, rather than all the features in wake, trying to look for those in REM sleep. So it's a much more sensitive measure. And when we did that, we indeed were able to classify And here's what the data looked like. We didn't have the temporal precision anymore because we had to use the whole trial to train during REM. But basically, here's the classification accuracy. So this is sleep time and wake time, sleep time since the cue, wake time since the cue. This is the area that classified above chance in the experimental night. You can see it's not there in the control night. And this is when we compared the two. So there's a nice kind of time period where we could tell which hand is being cued. And furthermore, the extent to which the strength of this AUC, the extent to which we could tell this, actually predicted the extent to which people got faster at producing that serial reaction time task across the night of sleep. So we thought that was really nice. I have to say we don't usually get this correlation, but we got it in this case, so we were super happy. So the answer is yes, the TMR definitely triggers the brain to practice the information, whether you apply it during non-REM or REM sleep. Okay, so what are the impacts of this? Is this just a kind of a quick phenomena where you get a brief improvement in performance and then it's gone within a couple of days? Well, no, the answer is no. Martina Rakowska, who was another PhD student in the lab, asked, what are the long-term effects of this on the brain and on behavior? And so she used that same task, the serial reaction time task, and she tested people not just the first day after the overnight manipulation. So they had one night of targeted memory reactivation, tested them the next day, at 10 days, and at 20 days. And here, what you can see is, so the reaction time, you want to be getting faster. So you want this reaction time to be getting lower. And so the red ones are the cued. And you can see that it is slightly lower at day 1 and day 10, but not significantly so. So if we did a main effect across all of these sessions, we do get a significant cueing effect. But if we look within each session, it's not significant at day 1 or at day 10, but it is significant at day 20. And so it looks like the impact of this behaviorally is actually continuing to unfold, at least out until 20 days. In other studies, we've looked a little bit further, and it does look like this fades away, and eventually you lose this. But certainly, it's in at least for this task, it looks like it's there until 20 days after. So it's not really a short-lived effect. What's going on in the brain? Well, Martina put her participants in the MRI scanner the day before, on day 1 and on day 10 when they did this task. And then she looked at how changes in brain activity between those sessions were predicted by the behavioral benefit. And it's a little bit of a complicated contrast here, but basically changes in the contrast of cued uncued. So they're doing the task in the scanner. We're comparing brain activity when they're doing a cued to an uncued sequence. And we're looking at how that changes and how that's predicted by this benefit at day 20. Nice kind of plasticity in terms of functional response in the sensory motor cortex. We also found structural changes, largely in the precuneus, actually, and some other areas, but I haven't put them on the slide. So the message here is, not only is it not just a short-term thing, but this one night of stimulation actually leads to long-term ongoing plasticity in both function during the task and structure of the brain. So triggering this reactivation is really doing something to the way the brain is handling that memory. And what we think in the lab at the moment is what it's doing is starting a process that then continues to unfold. So it's not just that your brain only practices that memory when we cued it, it's that that memory gets tagged and then it continues to be practiced again and again and again, probably during wake as well and during sleep. And that process just continues to unfold for quite a period of time. And how long that is probably depends on the individual. It probably depends on the task. It probably depends on how you cue it, but it's not just a one-time thing. Okay, so I'm going to shift gears now and talk a little bit about emotion. So back in 2009, Matt Walker and his colleagues introduced this idea of overnight therapy. And the idea here is that if you have a memory, an upsetting memory, where you would like to decouple the emotional content from the memory itself, one of the ways that that could happen is by reactivating that memory in a context where you're not having the same kind of upsetting bodily somatic responses. So things like skin conductance, heart rate responses, pupil dilation, your somatic markers of arousal, if those could be dampened down as you're reactivating the memory, then it might help you to decouple the memory itself from that response. And it turns out that REM sleep is just such a place because in REM sleep, the locus ceruleus stops releasing norepinephrine, and norepinephrine levels go to floor, and that is the neurotransmitter that mediates many of these bodily responses. And so you simply, at least in theory, you simply can't have those responses in REM sleep. So he proposed that if the brain is reactivating upsetting things in REM sleep, then this could be helping us to decouple the emotion from the memory. So one of my students, Isabel Hutchinson, got extremely interested in this because she said, well, if that's the case, then we might be able to harness this therapy by triggering these upsetting memories in REM sleep using targeted memory reactivation. So how did she do this? She used a very simple paradigm. She just used standard IUPS pictures, so upsetting pictures paired with semantically related sounds. She asked people to rate them on a 1 to 9 scale for how arousing they were. Then she had some checks to make sure that people had really learned the pairing with the sounds. Then people were wired up for EEG. They slept overnight in the lab, and we had 2 different groups. One group in whom we applied TMR in REM sleep, and another group in whom we applied it in slow wave sleep. And I should say her images, so half of her images were emotional, were negative, and half were neutral. And when we applied the TMR, we always reactivated half of the negative ones and half of the neutral ones, and we kept out the other half as controls. So we could check the impact of the TMR. Okay, so what did she find? So here you can see the overnight habituation. So that's the extent to which the arousal came down across the night. So, and it's normalized by baseline. So a higher bar means more habituation here. And what you can hopefully see is that there is a main effect of cueing, but that main effect is actually driven by the negative items. So it's quite a messy, big distribution for the neutral items. So if we just look at neutral, that's not significant. But if we look at negative, it is significant. For the slow wave sleep group where we applied the stimulation in slow wave sleep, there's a difference between neutral and negative for habituation, but there's no cueing effect at all. So this really only worked in REM sleep. Okay, so we got all excited about this in the lab, and we decided to follow it up with another study. So Viviana Greco, another PhD student, asked, what is this doing to the brain and also to your autonomic responses? And to look at this, she used almost exactly the same protocol as Isabel, except that now we got rid of the neutral images because it was messy and we were only really interested in the negative ones. We put people in the MR scanner after the 1 night of stimulation. So we didn't put them in the next day, but 48 hours later, basically in the evening. And we also monitored their heart rate. And so just in brief, what did we find? Well, in my view, this is, it's tremendous. We found a down regulation of the whole salience network. So here I'm showing amygdala, insula, orbital frontal cortex, but we also had, these were our regions of interest. But there was another area that was not a region of interest. So it's the dorso-medial prefrontal cortex. It wasn't a region of interest, but it survived whole brain correction. So we had to go and look up, what is that doing? And we found this is the key hub of the salience network. So basically, the whole salience network responded less when we showed people these negative images after they had been reactivated in REM sleep just for 1 night. We only reactivated them about 10 times. compared to if we hadn't reactivated to our control. So really strong MR results. In terms of the physiology, we also found a reduction in the heart rate deceleration. So when you see something that's upsetting, you have a brief slowing of your heart rate. That's called heart rate deceleration. And that response was reduced for the items that have been cued in REM sleep compared to the uncued. And we also essentially replicated the behavioral result from our earlier paper from Isabel, but we only replicated it in the items that were rated as quite upsetting at baseline. So this time we had a scale of 1 to 5 for how arousing things were, and it was only items that were rated 4 or 5 for arousal at baseline that showed this effect. The other ones didn't show an effect of cueing at all. So this is kind of, you know, still something that we're working on and analyzing, but we are quite excited about it because it seems that this cueing, not only does it lead people to subjectively say that things which they found arousing and upsetting are not as upsetting anymore 48 hours later, but it also leads to a change in their objective physiological responses and in the salience network. So off the back of that, we have a current big project going on where we're looking at how we can understand this kind of sleep engineering, which is really aimed at tackling negative emotions in order to think about, could we use this to tackle PTSD? So we're very early stages. We're only 6 months into this project, but I'll just tell you what we're doing here. So there's a risk that in PTSD, where we're talking about really traumatic experiences, this just won't work at all. But we hope that because it seems to be most effective on things that were rated as most arousing at baseline, there's a chance that it will. So it seems to us that it's worth a try. And so in this project, we're asking, how long do the benefits last? It's no good to us if it's gone in 2 weeks. You know, we need to see if this lasts for time. What does it do to the brain? So we'll be replicating this study, but also looking for structural plasticity. And also, how many nights should we stimulate? Because until now, we've only stimulated for 1 night in all of these studies. But we're developing a device that can be used in the home to provide targeted memory reactivation as a form of sleep engineering. And so we'll be able to stimulate for as many nights as we want. And so we can titrate that response and really see whether that would help us to shift some genuinely traumatic memories. So it's early days. All of our work for the next 3 years will be in healthy participants, and then we'll start doing some feasibility studies in patients if everything's looking good. There's another side to this project, which is about depression. And here we have quite a different idea. Here the idea is, well, we know that in depression, negative thoughts tend to dominate and people tend to ruminate and dwell on negative things. And we know from our own work and from our colleagues' work on targeted memory reactivation in non-REM sleep that we can selectively strengthen the memories that we target by triggering them to be practiced in non-REM sleep. And so our idea here is to strengthen the positive memories. So we're halfway through our first study on this. We have no pilot data, basically, but we are asking people to tell, you know, we get a bunch of their own autobiographical positive memories. We also show them positive videos, and then we reactivate those in non-REM sleep. And we will check whether we can strengthen those memories, hopefully boosting the positive and also blocking the negative memories from over-consolidating themselves spontaneously. So that's kind of what we're doing in the lab at the moment. I have more slides, but I feel like this might be a good place to stop and get people's comments, have a discussion. I think this will be enough to give you the flavor of sleep engineering, and then you can ask me about the other things if you want to. So thank you very much for your attention. And I'd just like to thank all the people in the lab who did all this work, because it definitely wasn't me, and also the current lab members who are with me at the moment. And thank you very much.
Wonderful. Thank you so much, Dr. Lewis, for talking to us about your research. If you guys have questions, please type them into the Q&A box at the bottom of your screen, and we'll do some Q&A. I'll start us off. I'm wondering, how do participants feel about this idea of sleep engineering? Do people think this is cool, I want to do it? Or people freaked out that you can manipulate their memories? Just what types of responses do you see from the public when you talk about this?
Yeah, I mean, I think people are fascinated. I think, I get asked a lot about eternal something of the spotless mind. And, there are lots of sci-fi kind of movies and scenarios that kind of touch on this, and people are aware of those. In general, I think people are excited. So we do have, because our current project that I just told you about is funded by the Wellcome Trust, they have a real emphasis on kind of public engagement and experts from the community. So just talking with people who are not part of academia, not part of your research team. If in our case, some of them have experience with PTSD and depression, but some of them don't. And just getting their feelings about things and running everything by them. And they're super. We've got a panel here and a panel in Colombia, actually, because we're collaborating with Colombia on this, and they're super excited. And they just, they want to be talking about it all the time.
That's awesome. Thanks. Any other questions for Dr. Lewis? Okay, we've got one coming in. How does blocking the negative memories coming work technically? How do we know it's an upsetting memory coming and blocking them? Thank you again.
I'm trying to understand the question. How do we know it's an upsetting memory blocking?
I think they're wondering kind of how would the brain know that the memory was upsetting to then not activate it?
So I don't think that that's the way it works. So if you're talking about the work that we're doing on depression, trying to block the brain from reactivating the upsetting memories. The idea is, so we know that this reactivation is kind of a limited resource. So it's not the case that your brain can just reactivate unlimited numbers of everything. We always see trade-offs. So if we reactivate, if we've got, say it's the finger tapping thing. So supposing we got two sequences, if we don't do anything, if we don't reactivate anything, then they'll both consolidate a bit over sleep. And that will be predicted by the time spent in slower sleep. But if we do reactivate one of them, then that one will get better, but the other one will actually improve less. So there's a kind of a cost to the thing that doesn't get reactivated, get triggered, because all of the resource we think is getting taken up by the one that we are triggering. So the idea with the positive memories is we are just going to blast the positive memories the whole time in non-REM sleep, and we're not going to leave any time, any opportunity for those negative memories to creep in. I mean, they probably will to some extent, but we're hoping that by kind of jamming the system with the positive stuff, they won't have so much opportunity to over consolidate.
Great. Yeah, I think that's a clear explanation of that. Okay. Whoa, lots of things just popped in. Okay, is the sound replay in REM rhythmic and synchronous?
No. So we try to avoid it being rhythmic. So what we are not trying to do with this is create some kind of a resonance, or boost a particular oscillation. So we usually jitter between the sounds. So there would be some kind of a random interval between the sounds. Other people have played with trying to use sounds to stimulate particular frequencies. And I think you can do that to an extent, but that's not what we're doing.
They've got a follow-up question, wondering if you could replay positive or negative melodies in REM as the sound cue that then might push around the benefits that you're seeking.
Yeah, potentially. just to be clear, so in REM, we are reactivating negative things because we're hoping that that will help to decouple the emotion, the negative emotion from the memory. With the positive, we're replaying those in non-REM because that's where we can strengthen things and we want to strengthen the positive. I think that you could do that with melodies. I think there are a few things that make it less optimal. I think one is just that if it's kind of an ongoing thing across time, the brain might just habituate. So we tend to want our stimuli to be kind of short and then a little break and then another stimulus. A colleague did try targeted memory activation just with playing music all night and they didn't get any benefit. So I think probably the brain just got used to it and filtered it. So I probably wouldn't go for a long melody, maybe something very, a few seconds. Yeah, maybe. The other thing is, my feeling is that people's own memories are more potently emotional than something that we choose externally, like a picture or a melody. So that's why we're trying to go for autobiographical memories these days. But that might vary between people.
Oh, so another question says, I know there's some evidence that sound REM sleep might help disarm negative memories, whereas restless REM sleep doesn't help or might exacerbate negative memories. Have you been thinking about that issue with your study with PTSD survivors?
Yeah, so that is something that we are worried about. So there's definitely data about that. Some nice papers out there showing that disrupted REM sleep doesn't have the same benefits. And also in people, even people who are just prone to PTSD, their REM sleep tends to be abnormal. So they tend not to have this reduction in norepinephrine to the same extent that people who are not prone to PTSD have. So it is a concern. But the reality is that they do still have REM sleep, and even if the sleep is a bit disrupted, it's still there. And noradrenaline is not the only hormone that's relevant for this. It's just the one that's kind of simplest to talk about. So we very much feel it's worth trying. It might not work, but it's like anything. We're going to give it a shot. First, really try and understand the system as well as we can in healthy people, and then we will try it in patients if it still looks promising, after ask me in 3 years. Yeah.
Oh, yeah, that's science. All right. I was curious in the acoustic simulation study you showed in young adults versus middle-aged adults, whether you think there are other variables or demographic factors other than age that might necessitate tweaking acoustic stimulation parameters to be effective, or whether personalizing acoustic simulation to each individual is kind of the future.
Yeah, I think absolutely. I mean, I think the more you can personalize these things, the better. So I'm not sure if the person asking this question is aware, but it's not available so much anymore, but there used to be a device called Dream that delivered these stimuli. You could buy it off the shelf. It delivered this quite nicely in home. And they, as a company, collected tons of data from people who had bought the device or from their own studies. And they really found a huge variability in effectiveness. So I think, in 25% of people, it worked amazingly. Another 25%, it just didn't work. And then you've got the 50% in between where it's kind of a bit hit and miss. Also, some people found it just really uncomfortable to have something like that on their head and they couldn't tolerate it. I think there are lots of factors, so many factors. Yeah. Definitely personalizing is going to make these things much more effective. But the great thing is that we have the technology now. It's absolutely out there to personalize all of this. So I think it's coming.
Great. All right, well, I think we will end there. As a reminder, our next One World Psychology speaker will be next month, April 23rd, where we'll have Alan Komen talking about large-scale social outcomes and psychological phenomena, measurement to interventions. But for today, I want to give a hearty thank you to Dr. Lewis for sharing her expertise with us, and a reminder that the video will be posted on the Psychonomics website in the next few days. It takes a little bit of time to be captioned, but then it will be posted and freely available. So thank you all again for attending, and we'll see you next time.
Thank you very much.